Hello everyone, welcome to Ash Jottings. This video lecture will talk about triple helix or HDNA. So let's get started. DNA is one of the most and intensively studied molecules, and scientists are still uncovering the layers of structural complexity. So as we all know, in the year 1953, James Watson and Francis Crick, they developed the hydrogen bonds that help DNA base pairing. After a decade, it was Carl Hoogsteen who recognized that bases had a potential to pair up in alternate way. That's interesting. Now, how they were able to do it? The purine bases, that is the adenine and guanine, would flip their normal conformation and form a new set of hydrogen bonds with the partners. So, what does it mean? What actually is that? Usually, if you talk about the DNA double helical structure, if you can see, it is double helical structure. Um, double helical structure. So at later on, what they found was in the major groove region, there existed one more strand which used to come like this. Okay, so instead of two strands, there were three strands. And how were they base pairing? They were base pairing like the purine, which is the adenine and guanine, where base usual case it is with the, for example, if you take it is adenine with thymine. And you know what happens in this case, that adenine can base pair with the thymine using the Watson and Crick base pairing. Apart from that, it can also base pair with another thymine using this Hoogsteen base pairing. Hoogsteen base pairing. In order to give it a triple edge kind of, triplex helix view. So let's move on to the next slide. Now, they need to prove it. So what did they do? Their study relied on indirect nuclear magnetic resonance data from the purine carbon atoms, which do not directly participate in the hydrogen bonding. So they actually developed a technique where they monitor the purines, the nitrogen atoms, which take part in the hydrogen bonding. So they took a short strand of DNA and they start monitoring the purine. And what they found is that there were certain signals which actually were occurring for certain nitrogen atoms were changing over time. So the after a specific time, there were signals which indicates that certain nitrogen atoms were changing over time. The changes which indicates that some nitrogen atoms have lost a hydrogen bond or while others have gained it. So when they actually saw it, when this experiment was conducted and they found out that, they saw that the pattern of binding is not Watson and Crick, but the pattern of binding was the Hoogsteen base pairing. A Hoogsteen base pairing is non-Watson-Crick hydrogen bonded motif observed in nucleic acid. The two nucleosides from two different nucleic strands or from distal regions. Okay, it may be from the different strands or from a distal region of the same strands were held together by the Hoogsteen base pairing. Now, two nucleobases, one of the each strand can be held together by the hydrogen bonds in the major group. So usually this will occur in the major groove. Okay. Now the base pair, how does the base pair occur? Let's see. The base pair applies N7 position of purine, which will be the hydrogen acceptor, and C6 amino group as the donor will go and bind to, will bind, or hydrogen bind bonding occurs with the N3 and C4 Watson-Crick surface of pyrimidine. So Hoogsteen, I will be explaining it using the structure so it will be more clear. So let's see the rest of it. The Hoogsteen base pair exists transiently in some DNA molecules and in thermal, which is in thermal equilibrium with standard Watson-Crick base pair. They're also observed in protein DNA complexes and also in the D quadraduplexes. It is actually hydrogen acceptor, and N6 is the donor. It is going to and Crick, and other one is the Hoogsteen, 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 okay, EN Steen base pairing. This is a 7, 8, and 9. Right. So usually the bonding will occur with N and in this. Okay, please. I'm not going to fill it all things because already I have spoken, I have taken the video regarding this. So let me just show you briefly of what happens. So it will be like, this will be the one, two. So this is time and so yeah, methyl group is present. So it's going to bind them all, right? It's going to share, and here it is NH. Okay, so this is how the binding usually takes place, right? Now, apart from that, what will happen, it will also share a binding directly from here and also with here. So in H1 more, right? It's going to base pair with so this will be the third and the fourth. Second, fifth, sixth. So we have the methyl group in the first position, and here you have the second bond. So this is the Watson and Crick base pairing, and this here, this is called the Hoogsteen base pairing. Okay, so this is what we were talking. Now that you are clear, now let's talk about what is the triple helix. Triple helix. So triple stranded structure, they can occur during recombination of DNA, recombination of DNA, or during DNA repair. So what actually happens in the triple helix is that the structure involves in the addition of normal Watson and Crick base pairing the Hoogsteen, as I told just mentioned. So it will have just by this short Watson and Crick plus the Hoogsteen base pairing, Hoogsteen base pair. BP, okay, right, BP. So it involves both this So, for example, you can say if a purine, where you can say, as I just showed, the adenine is going to base pair with thymine, with the Watson and Crick, it is going to base pair with another thymine using the Hoogsteen base pairing. So this type of triple base bonding is biologically very important. For instance, it can help to stabilize the structure of tRNAs. So this triple lex helix, and also known as the HDNA, is, just write it on here. This is the so-called HDNA, a portion of the, what actually happens, why is this mixing occurring? A portion of the pyrimidine and the purine duplex denature and loops back to form triple helix. That in, it is going to leave the one strand, leaving one of the strands from the denatured portions unpaired. Okay, so what actually happens, let me just draw it here. You have the phi dash and you have this 3 dash prime. Okay, so there will be an unwinding occurring. Okay, then you have a base. You have a base. Okay, you have this, the base coming. Okay, apart from the other strand. Okay, you have the base. Suppose this is the scenario, and consider this is your A, and this is your T, and this is A, and this is T. Suppose what happens, it is going to base pair this with this, okay, or this with this. Okay, so in this case, you can see if these three are actually base pairing with each other, it is leaving this strand, okay, which is left. That is what I mentioned, leaving one of the strand from the denature unpaired. So if these three are pairing, and it is going to leave this strand. Okay, so I hope you understood. So it usually Watson and Crick base pairing alone happens, but in the case of triple helix or the HDNA, there are two types of base pairing occurring, that is both Watson and Crick base pair, and then it has Hoogsteen base pairing. In in fact, co-crystallization of monomeric adenine and thymine derivatives invariably leads to Hoogsteen pairs, suggesting that this Hoogsteen base pairing is more stable for A base pairing with T than G base pairing with G base pairing with C. So it is most common, you can see it will be more common in A T base pair than in G C base pair, because here it has to be you know, it has to be just a minute. So what I was thinking, yeah, G, and this has to be protonation should occur in order for the cytosine to go for this kind of base pairing. Okay, now I hope this is clear. Let me just mention or summarize it all together. See, sometimes there is a triple helix or HDNA which is present, and in order to this can occur in when the recombination of DNA takes place or when there is a DNA repair, and also in few books I have seen when there is a very low pH, automatically this kind of structure can be seen. Okay, so what happens in triple helix? You have two kind of base pairing. One is the Watson and Crick, and another is the Hoogsteen base pairing. So it is like the purine, pyrimidine, and pyrimidine. So you can say you have purine, usually base pair with the pyrimidine, but here it can also base pair with another pyrimidine of the same strand or from a distant regions. Now it will what happens, as I mentioned, it will be seen like this. Once purine pyrimidine duplex denatures and loops, and this is getting denatured and loops back to form a triple helix structure. So how it happens? So what happens once it gets denatured? It is going to position itself here. So it is going to take up these two strands. This will be unpaired, and if it's going to take up these three strands, this will be unpaired. Okay, in that it is most commonly seen in adenine and thymine than in guanine and cytosine. That is because in the case of guanine and cytosine, the cytosine has to be protonated. So it is, I think it is very clear to you right now. I'll just give you a more clear. If this is a helical structure, okay, now what happens, it is going to unwind, okay, denature. Then what will happen? It's going to take up like this, right? mm, you have your double helix coming this round. What happens is this gets, it gets taken into form. Okay, so this will be unpaired. This is what happens. Okay, thank you.